Okay, so today I will be talking about flow cytometry sample acquisition and data analysis. So before going to do any data acquisition and flow cytometry data analysis, it is very important to uh, it is very important to prepare good quality of samples, because good quality of samples can give high quality data and very reproducible data. So the two important things for sample preparation that we should consider, it is that the cell viability should be very high, because if there are a lot of dead cells are present in the samples, dead cells can non-specifically bind to the staining reagent, here the antibodies or any fluorochrome conjugated reagents that can lead to high background signal and give false positive result. And the second important thing that we should consider to when we are preparing samples, that the samples should be in single cell suspension, because it can reduce the chance of flow cytometry clogging, and it also reduce the number of doublets that can give a false positive. It is important to process the samples when they are fresh, and do the staining as soon as possible when you get the samples. When we have blood samples, so we should pay attention that what kind of anticoagulant we are using, whether it is EDTase, sodium heparin, or acid citrate, because depending upon the anticoagulants, it can affect the expression pattern of the biomarkers that we want to explore. And blood sample, when we doing the blood sample analysis, so the blood sample should be processed as soon as possible, because delayed processing of blood sample, especially when we're doing high call density gradient purification, it can reduce the cell numbers, and it can also cause granulocytes contamination in the monolayer. And when we have solid tissues, we can make it single cell suspension using enzymatic digestion or mechanical dispersion. When we have bone marrow cells, we can just use syringes, different syringes with the needles, like 27 gauge, 27 and a half, or 26 gauge needles to make it single cell suspension. When we have the samples and then we are ready to stain the samples, we should so we should use the 5 ml tubes that 5 ml tubes that are recommended for flow cytometry data acquisition. And the biohazard sample should be placed in a box, and it should always be capped. The concentration of the number of cells that we want to acquire, it is important to have a good number of cells. The minimum number of cells that we should have per ml is at least 1 million cells. And the volume for sample acquisition is normally we use 250 to 300 microliters. For flow cytometry data analysis, we need different types of controls, and these are the 3 most important controls that we should use when we are doing flow cytometry data acquisition. The biological controls that are like the same cell type that we are staining for different biomarkers. So the biological control, that is like if you are doing bone marrow cells, so you should have unstained bone marrow cells as your biological control. Compensation controls, so that is the compensation controls that helps you to have the single, that helps you to get the to make it that whatever like to make the right gating and the compensation control. Suppose if you have 6 different fluorochromes, so you should have 6 compensation controls. For each control, each fluorochromes, you should have individual compensation control. And then you also should have a fluorescence -1 control. So depending upon how many tubes you have, how many antibodies you have, you should, based on that, you should have FMO controls. So if we are using 5 different antibodies, so we should have 5 different FMO controls, and each FMO control we should have 4 antibodies that we are using for our experiment, except that 1. So 4 antibodies in each tube, but each tube like 1 -1. So if we have Phe, PE, PE size 7, APC size 7. So the fluorescent FMO control 1, we should have Phe, PE, PE size 7, APC, but not APC size 7. So it's the same way we should make a compensation fluorescence controls for each of the fluorochrome that we are using. We normally acquire data on Fortesa, and this LSR Fortesa. This machine is equipped with 4 lasers that can acquire, quantitate at least 14 different fluorochromes, and the software that can it compatible with the LSR Fortesa is FACS-DIVA, and FACS-DIVA software can be used to acquire the data and even for the analysis of it. So when we go to the machine LSR Fortesa looks like this. So it has different buttons. So here that there is low speed, medium speed, high speed, and there is also that run, standby, and prime. It has different functions. So all the top one that define the speed, at what speed we want to acquire the samples. So how many events we want to acquire per second. And here that when we acquire the data, it is this left-hand side button that say that it is like when you check that, hit that button, it start acquiring the data. And the second one is the standby, and third one is the prime. So it has, when you don't want to acquire data, you can just hit standby. And prime is used for primarily to clean the probe. So when you are done with the experiment, we normally hit prime to clean, putting bleach here in the probe, and then so the probe gets cleaned. As I said that BD FACS-DIVA software are being used to acquire the data. So when we go first, we have to log in, and it has 2 computer. It is easy to to open several windows on the screen, and the same time we can have the acquisition sheet. So when we log in and on BD FACSDiva, we see this kind of window. There are several windows popped. The first one on the left-hand side, we have to first, if I click here, so first thing that we have to open a new experiment. So by clicking here, we can open new experiment. We can write down the name, whatever we want to put. So here I put that particular experiment that we were doing that day. It was NPY overexpression. And then the what kind of samples that this one is the samples that what kind of samples you were doing. So here we were doing bone marrow cells. So and then looking for hematopoietic stem and progenitor cells. So just look bone marrow HSPCs, and then followed by tubes. So when you just click when you log into FACSDiva, and then you have these windows on this left-hand side window, you just click this, then you will have this, it will open up here. You write down whatever your specific experiment you want to do, followed by you just click here, then it will give you the sample information. You can write down whatever you want. I normally write down the what type of the tissue I am using and what kind of particular phenotypic analysis I'm doing. So I put bone marrow HSPCs. And the tubes, you can put the particular what specific tube, what kind of sample is in that particular tube. So here that we had wild type my cells, and the NPY overexpressing mice derived cells. So it just depends at what way you want to write. So when you have you open new experiments, and then you write down the your sample name, and then you have to after that you can label the different antibodies you want to label, so that what kind of antibodies that you are using for that particular experiment. So you just click here on labels, and then this window will pop up, all this information will pop up, and then you just put suppose your FIPC is you have FIPC conjugated CD48 antibody, so you just write down CD48 for PCI 5.5 here is CD150. So just write down the information, the information of the antibody that you are using, so that you remember that what antibodies are conjugated with what flow. Then when you have here, then you write down this information here, and then you check here status. So when you have status, so then it will show up. Okay, before going to here, so when you have different commands here, and on that other screen that you can have the acquisition sheet. So on the acquisition sheet, you can here on the top, there is like different different commands that can you use to draw your the dot the dot plot profile or contour plot or density plot, depends upon which way you want to see your data. And once you have here that you have your the experiment information, your sample information here, and when you, and then here the status, when you click it here, what it will do that you will have a several information here, it will pop. And then when you click parameter, in parameter section, you just check whatever you want to have, what types of antibody that are conjugated or what types of fluorochrome, that you just check and see that how many fluorochromes you had for that particular experiment. Suppose here we had these antibodies that were conjugated with these fluorochromes. So we have these fluorochromes that are showing up here. But if you want to delete, you can delete just clicking whatever you don't want to have, or you so just click and then here delete. It will just disappear from the screen. If you want to add additional, so you can just click here and it will start showing up here. So once you have this, you know that based on your antibody information, you can either add or you can remove different fluorochromes. Then you have the voltage. And so the before going to the voltage part, here there's like there you can see that there are different boxes that are checked. So when you want to acquire the data, so here the FSC, and this is FSCs that define the cell size, and the SSCs that define the cell granularity, how complex the cells are. So what normally FSC is almost I always use in linear scale. So this should be unchecked because all other are checked. This is log scale. But if you want to acquire data for FSC, the FSC setting you want to have in linear scale. So please just uncheck this setting. And in addition to this, if there is some doublet, if there is some cells that combine together, stick together, so to exclude those cells, you should check here the area and height and width. All three, these boxes should be checked. That will later on help you to exclude the doublet cells. So that and for SSCs, I normally use log scale, but some people they use linear scale. It is just personal choice that, and it also important, and also that what type of experiments you are doing. So depending upon your experiments and and also your choice, which one you like more. So you can either SSCs, you can do either linear or log, but FSCs almost always it is in linear scale. For for fluorochromes, like all these fluorochromes that we have, so normally almost 99% we do in log scale. So we have to check these boxes that so that it can acquire data in the log format. And but there are some exceptions. If we have we are doing cell cycling, so in that condition we should just uncheck this box. Suppose we are using DAPI. So DAPI, it is similar to Pacific Blue. So we have to uncheck this log box and it should be just linear. And then when you want to acquire the data, so suppose you start with your unstained sample, you should just put the here, yes, here load your sample and then click here start, run, and then it will start taking samples and then hit acquire. So when you are acquiring the data, it will, when you are acquiring the data, before acquire the data, you should plot here all the possible fluorochromes that you are using. So if and in addition to that, you should also plot FSCs and SSCs plot. So on this acquisition sheet, you plot FSCs, like I normally plot FSCs on y-axis and SSCs on x-axis. And then I plot for FSC's area versus height to exclude the dead cells. And then I plot dot plot for my all the possible fluorochromes that I'm using. So I'll start with suppose if I have a Ph.D. and PE that day. So I will just plot just Ph.D. here and PE on the y-axis, but we can do other way around also. So if I have only one color, that's is because you just need to plot one dot plot. But if you have several colors, suppose you have 5 different colors, and so you want to see that like whether the voltage that you are putting here are those voltages are good for these samples. Means like when we put the samples here, so the cell should be here. The cells should be like above 0 on both sides, x and y axis, but it should not go outside the plot area. I will explain you later on, just showing a figure. But that's the way we start. We plot different dot plots, contour plots, density plots, and depending upon what we like. And then we just run the samples and see that whether the sample that we are running, it is in this area. It should above the 0 for both the axes, but if it is voltage is very high, so it will go out of the plot, and in that case you have to reduce the voltage. Suppose 50. So if the 50 voltage is very high, it will, and this is our x-axis defined the 50 antibody, Ph.D. conjugated antibody, so it will come out of this plot. So you have to reduce the voltage for Ph.D. And the same for all different fluorochromes. That we need to do that. All the antibodies that we are using, the plot should be should above 0, but it should not go out of the plot area. So we use voltage to fix these things, and and this is very important to do during the data acquisition. We should we should make sure the we are putting the right voltage, because if the voltage is not right, we can't fix that later on. Once the data is acquired, we can't do later on unless like we rerun the sample again or redo the whole experiment again. If we don't realize during the time of data acquisition. So this is the like all the used by you can just click here and you can increase or decrease the voltage or FSC voltage for the fluorochromes as well as for FSCs and SSCs. And here these are the commands that you can use to acquire the data. Suppose here that you just when you click here, it will start acquiring the data. When you see click here, it is into acquiring the data. It will also record the data. And here, these boxes showing that whether you want to acquire all the events that are showing here, or you want to have a particular gate. Suppose if I want to acquire only P1 gate data, so I can change here that the event to record. I can just here I can click and say that P1 gate, and then event to display. that what event that which one I want to show on the screen. So if I just want to display, want to see P1 gate, I can just click here, and they say that okay, that then you start only showing P1 gate. And here that how number to records. So how many events we want to record. That totally depends upon that your cell population. If your cell, your population of your interest is like in good proportion, so then you can give, even if you are requiring 100,000 cells, it would be good. But if your cell of interest, they are rare, then it have to acquire more events. Like for SLAM LSK that are highly enriched for hematopoietic stem and progenitor cells, and we normally acquire minimum 0.5 million cells, and try to acquire 1 million cells if we have enough cells. And this one is event to record, and this one just this is event to display. That how many events you want to display on the screen. These are the information, and so these three boxes that acquire, record, and then if you want to acquire that a specific population, and then how many events you want to acquire. So these are the important things that we should pay attention when we are acquiring the data, because we can't fix these things later on. We have to make sure that how many, that what we should have idea that how many, what is the frequency of our cell of our interest, so how many cells we should acquire, and then the voltage also, that I just said that when you're running the samples, it should be that it should just in this plot, it should not go out of the plot, and if it is, so you have to change the voltage for these things. If the cells are here, also if it is voltage is too high, then cells will go out of this above the this FSC axis, and then you have to reduce the voltage for that. So it is like voltage and number of cells that you want to acquire, and the cell population that you want to acquire. These things you can't do post acquisition. Once you are done, that's like you can't change this later on. So these things we have to pay attention when we are acquiring the data. And so for the compensation, till here that we have just like the different fluorochrome, the voltage, whether it should be in linear scale or log scale. But if you want to see the compensation information, so you click here. Then it will show up this this additional column, and this column can be used to fix the compensation issue. So all these windows that I've been showing, these are like I took this information from LSR Fortesa, but it irrespective of what flow cytometer we are using, certain criteria that we should those are very crucial, and we should always pay attention when we are doing flow cytometry data acquisition. And these are like select the parameter of measurement. So when you have that, this like what parameter you want to explore that day, you want to save your data that day, and the area width and height of different parameter. Like here, you have to make sure that which one you want. You want to have in log scale, you want to be in linear scale, and you want to for FSC that defines the cell size. To exclude the doublet, you you should check the area and height box. And, and then we have to set the voltage to make sure that it is all the cells are above the zero axis on both axes, and the cells are not out of the scale. As I said that here, that the select log scale or linear scale depending upon what you want to explore. Most of the time for the fluorochromes, it is log scale, except for the cell cycle. And select and adjust the threshold. Here is the threshold. So what it does that it helps to exclude the debris. If there is if you check here, if you click this option, then you will see there is it will pop up that the information, and there it normally the machine is set at 5,000. You can increase or decrease that threshold to either include the smaller size cells, or if you have a lot of debris and you want to exclude those cells or platelets, you can increase the threshold. And so the compensation part, these things can't be fixed post-acquisition, but the compensation part, it can be fixed post-acquisition when we are analyzing the data using Flowzo. As long as we have all the single color controls and unstained controls, so we can do that compensation. If we if we notice that there is some issue with compensation, although we have already done the compensation during the data acquisition, we can do it later on using Flowzo software. As I will just discussed that the scaling consideration. So our disclosure should be always linear. 90%, like most time people use linear scale. Side scatter can be linear or log scale, except that if there is like very tiny particles, or there is like nuclei, then the forward scatter can be in log scale. And the fluorescence parameter, that should be in log scale, except that if we are doing cell cycle. The reason to do cell cycle in a linear scale instead of log scale, because the difference is very little. If there is a cell division, and then we will only get twofold increase in fluorescent intensity, and that's why when we are doing cell cycle, we should do it in linear scale. FSC and SS is setting. As I said that when you were setting the voltage, so here, so how much voltage you should put, that is, it depends upon that what is the size of your cells and what is the granularity of your cells for FSCs and SSCs. So here, the voltage should be set in such a way that all the cells are within the plot. So it should not go out of the plot. But the same time, it should be also, if it is too low, then it will be very difficult to exclude the debris from the real cell. Like here, this is an example. So here, it is very difficult to identify where the real cells are and where the debris are, whether all these are like real cells, just because we put so low voltage. That's why everything is just like at the bottom, or it is really very few live cells are here, and these are just debris. So, so voltage should be in such a way that for the FSCs and SSCs, it should be in such a way that the most of the cells are in the center. So if the voltage for FSC is so low, it will be the situation will be like this. Everything is here. If the voltage is very high, so that the cells will start coming out of the scale, and it will be like, so you will see this kind of profile. So, and if you have this kind of profile, then if you want to see the singlet cell, it will be difficult to get that information. So the good strategy to set a voltage for the FSCs and SSC is this. The cells, most of the cells are in the center. You can see the debris here. You can see healthy and cells most of in the center, and there are very hardly any cells close to the axis. So this is the correct voltage. And for same kind of strategy, we should also apply for SSC. So cells, most of the for FSC and SSC, we should do in such a way that the cells are in the center. And same criteria we should use when we set the voltage for the different fluorochrome, conjugated antibodies or fluorochrome labeled reagents. So here, if in this case, it is the voltage for ABC is too high. So there's a lot of cells are out of is close to the axis. And here the Ph.D voltage is too high. So the 71% cells are sticking here to the axis. It is coming out of axis. So it is very important to pay attention on these voltage setting, that so cells should be not should not be close to the axis or out of the axis. And the best strategy for fluorochromes voltage setting is that it should be above the zero axis on both sides, and should not come out of the axis. The second important thing is, once we have a nice voltage setting for the FSCs, SSCs, different fluorochromes, then we can do compensation. And the compensation part we can also do post-acquisition, but I prefer to do compensation during the sample acquisition. So what is compensation? What does really compensation do? So it corrects the fluorescence spillover. So if suppose here Ph.D, so if the Ph.D, the emission spectra for Ph.D conjugated Ph.D fluorochrome, it should only give only emission for one particular, it should be excited by one particular laser, and then it should give it should have a emission spectra that can that is, if it's detected by only one particular detector, and it should not have any other emission spectra. But in reality, most of the fluorochrome, they have like it is a spectra, not necessarily only one particular wavelength that at that wavelength it emits. So in case of Ph.D, it can it has emission spectra that is specific to Ph.D channel, but in addition to that, it also can give signals in PE channel. So when you have a single color single color controls, Here that I'm trying to define here that what is the function of single color controls. Single color controls, it just helps to set the compensation. If this in your single color control, suppose in FITC, so in that particular tube, you have only samples that are labeled with FITC conjugated antibody. So the signal, when you run the samples, it should only give signal to the FITC channel. But here, it is not the case. It is also giving signal in PE channel. So how you can fix this problem? You can fix this problem by subtracting that whatever that spillover that is happening in PE, you subtract that value. So when you do the compensation, and then in that case, it will only give signal to the FITC channel, but not in PA. So here, if you see that if it is 872 events that are double positive for FITC as well as PA, but when we do compensation, this tube had only one antibody, so it should give signal in only one channel. So here it is when you compensate, so it is only giving positive signal for FITC, but not for PE. So how you can, when you are running the samples, how you can do this? You have this compensation. If you check this box, and simple way to remember that, suppose here FITC. So the percent, you just whatever on your right hand side, your right hand side, you should have the fluorochrome of your interest. So here it is FITC. So you have to subtract this FITC from the PE. So whatever that whatever the value that when you are putting here, it suppose here, like when I started putting number, start with 0.5, 1, 2, 3, 4, and then this will start going down. And when it reached to this point, then I will stop doing that compensation fix. So, so this is the way we do, and to correct the fluorescence spillover, and that's why it is very important to have the unstained control as well as the single color controls. Single color controls, it helps to set the compensation. And this is the way to remember. Just you, whatever fluorochrome you are trying to do compensation, that is on your right hand side, and then the fluorochrome against which you want to fix it. So here it is PE. So you just you pay attention here, and then start subtracting the values PE minus FITC, and you can just put here, you can increase or decrease that, increase or decrease the number here, and it will show up that how the population is changing. And then this is an example that where in for stem cells when we do. So here in this particular tube, that we had only one color, that APC conjugated c-kit antibody. So when after compensation, it is only giving signal here, but no signal over here. If it was, this is a very good compensation, that this is your negative and your positive. This is all the cells are aligned. If it is this, it is down, it means like it is compensated, and if it is up, so it means like you have to do compensation. And this one, this is in this particular tube, we had antibody for c-kit positive APC, c-kit conjugated APC, with and here that SCAR1 conjugated with the PE-Cy7. So after compensation, it is only giving signal in PE-Cy7 channel, but not in c-KIT channel. So not in APC channel. In addition to the single color controls that fix the spillover issues, and there is another problem that is like when when you are all the antibodies in your one tube. So in your samples, you have all the antibodies. So there are like some antibodies that are if the very bright, and there is that is it can over it can bleed over in other channels also, and it can give us some false positive or false negative. In this case, the FMO, the fluorescence -1, it helps to determine where to set the gate. In suppose like if we have just unstained, so in unstained that you think, okay, this is the this might be a place where I should set the gate. But when you have FMO, so in this case, this particular on this particular sample, and so it has all the antibodies except PE-Cy5 conjugated antibody. So if you compare to here, the positive, and it is like in brightness, it's significantly increased as compared to this. So if we make our gate based on this, then it will we will set our gate here. Most likely we will set our gate here. But when we have FMO, so based on this, we will set our gate here. So FMO controls are like better control to decide the gates for your different subpopulations. Once you have set the voltage, you have decided that how many events you want to acquire, and then you have also run all the samples, single color controls and FMOs. Then you can start running your samples, real biological samples that are labeled with different antibodies. Here I am giving an example of our HS species analysis, that are SS species analysis in mouse. We use SLAM LSK that are lineage negative, SCAR1 positive, c-KIT positive, CD48 negative, and CD150 positive. So when we have these samples and want to see that how this all the setting that we have done so far, whether it is good for these samples or not. So when we run the samples, I normally want to see that how the profile looks like during the acquisition. So I put all the gating normally when we do during the real analysis. So here, so I put FSCs against HSCs, SSCs, and so this is like the size and granularity. If the cells are in the inside the axis, it is not that too much over the axis, or like it is like very nice spread. So based on, we gated these cells as lymphocytes, and then after that I gated this particular population to see to exclude doublets. So if there is the two cells combined together, it can give you like a higher FSCA area. So when if the cells are single cell suspension, so all the cells should be here diagonal axis, and so we can get to that particular or the cells that are sitting at this axis, diagonal axis, and so we can get this, and after that we can further getting to like here the stem cells are negative for lineage markers. So all the based on the unstained and then FMOs, this is the by lineage negative population, and then we get it from here to c-KIT and SCAR1, and and we were interested in double positive cells. So you can see that very nice separation of negative and positive c-KIT negative cells and c-KIT positive cells, and same here for SCAR1 negative and SCAR1 positive. We were interested in LSK, so we get it here, and from here to further enrich our population, we added CD48 and CD150. So our population is lineage negative, c-KIT positive, SCAR1 positive, CD150 positive, CD48 negative. So the purpose of showing this these plots is to see that like all these like voltages and the compensation, they look good. We should when we are acquiring the data should even during the acquisition, if we plot the different population, we can get some idea that how good our staining is, how good our samples are. So once we start running the samples, we suppose once we are when we start sample number 1. So when we are done here, then once and we set that, okay, suppose I want to acquire 1 billion cells. Once we are done, then we go to the next sample and just click next, and that in that particular command, and then you acquire the next sample. And once you have finished acquiring all the data, you can export the data by clicking here. That your experiment, you click there and then export your data in FSC 3.0, because almost all the analysis tool that they are compatible with the FSC 3.0. So export the data, save your data in FSC 3.0, and also save the template. The template, it helps to if you want to use the same setting, you want to do similar staining, so you can just next time when you go to analyze your data, you can import that template and use the same setting for next time analysis. But when you are even like if you have saved the template and you want to reuse it, still it is important to you have all the single color controls, you have FMOs, you have unstained, so you can use the same setting but run the sample again, and so that make sure that okay, all the setting that you have done previous time, that's still working for the current samples. And so this is up to this, it was like how to set the voltage, how to set compensation, how to set threshold, and how to set the number of events that we want to acquire. and how to save the data. Once you have data, then you can use software to analyze the data. So normally we use Flojo software and to analyze the data. And for analysis, that like how you want to see your plot. So there are several ways um, to um, to plot your data. Histogram, dot plot, density plots, contour plot. It is like when you're plotting your data, it is important to know that with what the plot that you want to use, whether it is, and the subpopulation that you want to analyze, whether it is like related to your hypothesis. Like here, I can give you an example from here. Okay, when we plotted this way, we wanted to know that how many LSK or SLAM LSK are present in the mouse bone marrow sample. How does NPY overexpression impact their number? So we have just plotted this way to see that information. And so depending upon that, what kind of information you want to get, you have to use different plots. There is histogram, that is like sometime people use the histogram, and then sometime dot plots, contour plots. So histogram, it just shows the distribution of values for a specific parameter, like suppose secant. So here only I can see one parameter, the how many cells that are like secant positive and what is their intensity. And but it can't tell you about the relationship between two populations. It can't say anything about that how many cells of this secant positive cells are also positive for SCA1 or for any other markers. It can also, when you have this plot, it can also miss subpopulation that have similar values in one parameter. The second way of plotting this, so your data is dot plot, and so it is here it is a dot plot or contour plot or density plot. All these three types of plots are, but they are all bivariant. Means like you can see two variants at the same time. In this plot, you can this plot is showing the information about secant as well as the SCA1. So it can you can see relationship between the markers. When you are acquiring, you have already acquired the data. When you are analyzing the data, so there are like these these are the important important factors that we should parameters that we should pay attention. So the basic gating strategy, the way we should gate our cells for the analysis. So when you gate, so just gate the live cells, gate only cells, not the debris, like here. Like here. So here, that because these were the sample mouse bone marrow samples, and when we used mortar and pestle to take out the bone marrow cells from the bones. So there are like the bone cell, the bone parts also, when we crack the bones, the bone particles also gain. And so there is like here, there are like or like other like some debris that also can come to your sample. So you want to just get the cells, not the other debris or any like the bone part. So that's very important to pay attention that when you are gating. So gate only the cells, and then gate the single cells, remove the doublets, and then third it is gate the live cells. So first you get only cells, exclude the debris, and then after that you get rid of the doublets, and then get the live cells. So I can even here I can give you an example that here we excluded all the debris. Then after this we gated, and then this like the cells that are at the diagonally on for FSC area versus height, only those are the real single cells. So we gated this, and from here we wanted to have the live cells. So live lineage negative cells. So we did our live reagent, live cell detection reagent. We put with the lineage cocktail. So here by this gating, we excluded the dead cells as well as the lineage positive cells. So when we are doing this kind of gating strategy, you will get the first the real cells. You will exclude the doublets, and you will also get the live cells. When if these factors, if you don't pay attention about these factors, they can lead to a false positivity, because the dead cells, it can non-specifically bind to antibodies and give false positive signal. Doublet, it can have higher autofluorescence that can give a false positive, and if there are like debris, that can also non-specifically bind and give false positive. And in addition, debris can also soak up the antibodies, so that can also reduce the availability of antibodies. And I have already talked about the gating controls here, that you should have when you are deciding that where you want to set your gates for different for your samples. So you first start with unstained control. That unstained control, it will give you information about the background and autofluorescence. And then after that, for your gating for your different cell population, you should ideally you should use FM, and it can show you the background, and that how much background coming from the other fluorochrome conjugated antibodies. It can give you idea about the spillover. And then for the if you can arrange, you should also have positive control. Then it can so if you have a positive control, that like I already know that that sample should behave this way. So it will give you information about that your experimental samples, that whether they are giving the right result or there is some issues. And if it is also important, if you can have other biological controls, means like a stimulated cells. If you are working, you want to see that whether particular signals are affected by certain treatments, or particular markers affected by certain treatment. If you have some known controls and then known treatment that can stimulate the cells, you can also have include those controls. But these two are the very important controls: unstained controls, your and your fluorescence -1 controls for deciding your gating. You should also have the single color controls. That is that helps you for the just like the setting the initial compensation. So these these three controls: unrestrained controls, fluorescence -1 control, these are for your analysis, and the single color controls, that is for your compensation. Those are necessary for any flow cytometry data analysis. And I think that, can I stop right now? Hello? Hello. And I think that we are already just 2:10. So maybe that these are the criteria when you are analyzing the data. In addition to when you're analyzing, you should uh, decide where you want to, like whether you want to scale the plots. So there are like way in the Flojo software you can do that. And then when you have your data, the data can be the results can be shown as the frequency or the or like actual number of cells by multiplying the frequency with the total cell count, or if you want to see the receptor expression on particular cell type, the or receptor expression on cell surface, so that what is whether it is increasing decreasing, you should use the mean fluorescent intensity. uh, and So these are the two primary way to get the statical um, data, the frequency and flow cytometry. So I think that I would I can stop here. Okay. Do you have any? Does anybody have any question?